Hi everyone, this is just a talk I provided for the Rice Bioscience Welcome Symposium. And here I'm just recording and show people who are interested in my lab to say what do we do and then what can be done in the future. And then all of the work presented here has been generally published or already put onto our bioarchive. So for whoever is interested in the lab, please do reach out. However, I hope this talk just gave you a general idea on how the things progressing in the protein design field, and then if you are interested in the things we do. So first of all, what is protein design? Generally for protein design, we start from a function which, based on some doctor or biologist research, we know realizing this function would help on therapeutic development, etc. For example, you inhibit a receptor or you design an enzyme. And then for protein designers, you will formulate this function into actual interaction that will help to achieve those functions. The special thing we do is we use computation to achieve those desired functions, the interactions. Of course, since we're doing protein design, so we have the privilege of testing everything we design in the form of protein sequences. We, in the end, hope to actually put them into the desired condition to show those interactions actually did give you the desired function. This is actually pretty similar to what traditional protein engineering do. The major difference is we do it computationally. However, this changed a lot of things because generally for protein engineering, you're literally looking for a valley on a really big energy landscape. Traditionally, you probably have to do lots of experiments and then generate like a few hundred data and then figure out what to do next. However, using protein design, we use computation to significantly fasten the screening speed so that we can actually achieve one thing, which is making the previously unengineerable problem engineerable. Just give you folks a little bit example on what has been achieved during this point. There is success examples on enzyme design. There is nanobody and antibody design, and also commercially, there are also a lot of successful stories, such as there's design of cyclic peptide to target the cell targets inside the cell for therapeutic treatment. And then we can also design proteins which can sense the presence of biomarkers such as toxic protein of COVID, and then send out signals. This would, in the end, actually made it to paper-based COVID antigen detection. And of course, most recently, one thing is people design novel protein nanocages and then display multiple copy of antigen on the cages. This was, in the end, pretty successfully developed into a COVID vaccine. So for the problem I'm interested in, they are majorly focused on small molecule protein interaction type of problems. Generally, the problem is you start with a ligand, and then with all the different type of like computational operations, you generate a protein from scratch, have specific interaction affinity between your protein and the ligand. This is really important because this is the first step and actually fundamental step for you to generate protein binder, protein sensor for ligand, and also importantly, enzymes. So in this talk, I will majorly cover binder and sensor type of progresses. In my lab, all of the three directions are well covered. And then for people who are interested, we can actually use protein design to solve some of the ligand probing and also enzyme engineering problems. Okay, so one of the questions we want to explain to people is why is this problem actually also pretty hard design problem? The major challenge is, first of all, this is a really large sampling space because you have to consider the conformation change of the ligand and also the creation of the protein itself. However, this is not only a difficult and large sampling space problem. This is also a difficult sampling problem because you require really high accuracy. Generally, with protein interactions with ligand, you have to make what's so-called as short-range interactions, such as polar interactions, pi-axis interactions. Those means you have to be really, really specific and accurate when you are creating this protein. Data suggested your protein have to be as accurate as to 0.5 angstrom or even better to actually generate any binding affinity. For the past few years, I have been leading and developing machine learning-based method to generate binders between protein and ligand. We were able to generate binders towards critical metabolites such as cholic acid, larger ligands such as cyclic peptide, and also, more importantly, polar and flexible ligands such as methyltrypsin and stroxin. These really represent we are becoming more and more mature on our protein design abilities, and then actually can start to tackle more realistic and even challenging problems. So let me quickly go through how does protein design work for small molecule binder generation. So first of all, we sample the conformation of the ligand. In this step, we do not really do anything fancy. As long as the ligand conformation is physically allowed, we will be allowed this to be used at the starting point. That's to say, for example, for enzyme design, pretty often your ligand conformation or even your catalytic residue conformation is probably not the lowest energy conformation. That's going to be okay for this pipeline. And that is why this whole design procedure is acceptable for enzyme generation. Then we do something called scaffold generation, meaning based on this ligand, we want to design a protein scaffold have a height shape complementary to this small molecule. So just to mention, the scaffold here meaning is actually paying more attention to the CCANO coordinates of the protein. There is pseudo-residual sequences assigned to the protein. However, those residues does not matter, meaning you do not need to care about the side chains. So this step is called conditional scaffold generation step. In this talk, I will mention AlphaFold2-based hallucination method and CRF diffusion method that developed for this particular step. And then afterwards, we can enumerate the docking conformation between ligand and protein to start improving the docking poses of the ligand. This is pretty similar to the docking problem, and you can use the docking traditionally use energy-based docking method, and also of course some of the machine learning-based methods such as AlphaFold3 or Placer, etc. deal with this problem. And lastly, once you land into some docking method you like, you can assign sequences to those residues. So basically, only at this step, we start to care about the set chain information of the residues. This step is called fixed backbone sequence design problem. And then there is the element of the protein MPN and ligand MPN, which I will talk about. In the real design campaign, we try to enumerate between docking conformation and sequence conformation to converge into one sequence which achieve the physical conformation. So at this point, you probably realize there's actually two types of space we are iterating from. One is the coordinate space of the protein backbone. The other is a sequence space of the protein sequence. In the future of the method, we'll really try to merge those two spaces. And then the major reason of the spaces two spaces be like separated at this point remain more technical related. And then we imagine there are many of the model improvement coming from by either simply improving the searching of the individual spaces or coupling two of those spaces at the same time. So now let's still focus on individual steps. First of all, the structural space searching. No matter whether it's hallucination type of method or diffusion type of method, the design actually went pretty straightforward as you start from the ligand. You either pre-generate different pocket containing proteins and then using fitness of the protein and the ligand to check which is the scaffold have the best shape complementary to your ligand. Or you directly generate a scaffold around your ligand, which will directly have a good shape complementary to your ligand. So generally, identify the protein structure with or without docking are with this way are shared between two different types of scaffold generation method. So now let's first of all talk about hallucination type of method. So back in 2023, we think if we have a protein which have a pocket inside, then we can just using docking to check if this ligand is having the fitness to the pocket. Then we can turn this problem into how do we effectively generate all the proteins with pocket inside. We end up deciding we are going to generate C-symmetry protein, which will just have a pocket inside. In the end, we were able to generate AlphaFold2 based hallucination method to effectively sample all the protein with pocket. We first of all initiate the protein sequence with different individual unit sequences and some symmetric modules, such as repeatedness of 3, as shown here. Then we pass it to a structural prediction script such as AlphaFold2, and then this is the oracle that can generate the sequence and structure relationship using prediction. Then we wrote something called losses. This loss will check whether the NC terminus of the protein will have a close distance, and also the geometry of the protein if it's actually forming a C-symmetry. Then we wrote an optimization algorithm to really optimize based on this loss, and then make mutations of the original sequences. Pretty rapidly, within several steps, we were able to generate convergence of the structure based on the structure prediction model. And then all the coming up structure will have generally, first of all, a relatively agreeable pocket geometry, and also a pretty high folding score based on the structure prediction model. Because at this stage, the sequence is still with symmetry. Most ligand we don't want to bind doesn't really have symmetry. So we redesign the sequence using NPN to make the sequence more agreeable, and then no longer have the symmetry. So in the end, we have the structure with some sort of symmetry, and the sequence do not have the symmetry. However, the structure does still have the pocket inside. Because we do this on computer, the advantage is you can do it really fastly for many protein generations. That's how we generate many of the pocket containing protein, of beta sheets containing protein, alpha beta protein, and only alpha protein. In the end, we were able to sample almost 10,000 different scaffolds with pocket inside, and then use them to use docking to test which pocket fit the ligand best, and then continue form design from there. Of course, more recently, we not only can just pre-generate the protein, pre-generate the scaffold, and then using docking to figure out which scaffold fit the best with the ligand. We can also directly generate the protein with the ligand. I will talk about that in the later part of the talk. So now, then go to the problem of, let's say if you have a dog you really like, how do you generate the sequence of this protein? That's what we generated a machine learning model called Linna An. So in machine learning field, there is a field called graph neural network. So in this type of neural network, the data can be represented in terms of graphs with nodes and edges. The interesting part of protein and ligands is and then for the graph neural network, the nodes and edges, they can work like this. You can assign something as nodes and the relationship between atoms as edges. So the good part of protein and ligands is they can be naturally adopting this graph neural network structure. So for example, in ligand NPN, the residues are represented as nodes. The ligand heavy atom were also represented as nodes. The connection of the residues are represented as edges. And then, of course, the connection of the ligands and different bond type are also represented as the edges. This is good because then you can, first of all, add different information to the nodes and edges. For example, the ligand element type, etc. They can be added as features to the nodes. And then plugging into a pretty classical encoder, decoder sort of architecture of the machines, this allows the information to flow between the nodes and edges. And then the ligand NPN was able to learn the relationship between protein coordinates versus the protein amino acid sequences. Once the model got trained, we can use it for protein design. What happens is with the providing of the protein backbone coordinates and also the ligand docking conformation, it literally just returns the possibility of each type of amino acids at each position. Then based on this probability, then you can select which residue should be packed at which position. Based on the benchmark, we took some native small molecule binding proteins and then just designed those residues as and then checking if the ligand sequence design software can actually recover good residues around this ligand. We compare the results with traditional energy-based methods such as Rosetta and non-ligand aware machine learning methods such as protein MPN. We're able to see nicely ligand MPN have a better sequence recovery rate comparing with previous two methods, meaning it have a better sense of the ligand protein interactions compared with previous method. This ligand MPN was significantly improved the protein ligand interaction design, which is really helpful for increasing the success rate of interaction design. Then once you got designs, one question is like, how do you test them? Pretty often at this point, our success rate between ligand and protein range between 0.1% to 5%. For example, for some harder ligand, you probably need to want to test a few thousand designs to actually identify binders. In those cases, of course, you don't want to test them individually. What we do is we order those designs as oligo pools and express them on the surface of yeast. They will have a C-terminus MIC tag so that you can stain the presence of the tag using antibody fluorophore conjugations so that you can see, okay, there is this fluorophore signal, you know your protein is expressed. On the other hand, for the ligands, pretty often we have a fluorophore conjugated as well. So based on the double positive signal, you will know there is presence of the binder. So that's to say the workflow commonly look like this for the more harder ligands. You order a few thousand designs and then the oligo pool comes within 1 week. And then you need to prepare the yeast surface display library probably around 1 week. And then you do high throughput assays using cell sorters. You're looking for these double positive binding peaks. And then you isolate these binding populations and then using next generation sequencing to identify which one is your hits and purify the protein and verify through performing binding and purification assays. Parallelly, on the other hand, some of the easier targets we have pretty good success rates, such as 5 or even higher percentile, meaning you probably should just stick with like testing 50 to 100 designs, which can be done within plate scale. So generally, then the workflow can be even faster. You can order genes to be synthesized within 4 to 6 days, and then do transformation and golden gate assembling so that you can obtain the assembled plasmid within 96 well format overnight. With high throughput, with mid throughput expression within 2 days, then directly you can do purification and verification. So within 2 to 3 weeks, you can directly identify which are the binder and what's the binding affinity. All this can be done is under really one hood is de novo proteins generally have really high stability and doesn't really have much expression issue at all. It's really hard to imagine how do you do that for native proteins because most of them, they doesn't behave well and then doesn't really like this small scale type of expression and testing. So that's to say, de novo protein really help us to simplify the biochemistry characterization and wet lab experiments we need to do, and then allow us to focusing on the biochemistry questions we're asking for. Of course, just let you say some of the design results. We were able to get like nanomolar binders, and also micromolar binder to more polar and hard ligand, and some nanomolar binder to some polar ligands as well. From there, we also didn't really stop, as I mentioned, that I will tell you more advanced like scaffold generation method. Back in 2020, roughly, in the image generation field, there is really a wave of technology progressing that people realize you can introduce like diffusion-based architecture so that you can on your training data size establish a noise and pixel distribution relationship. And then reversely, you can slowly denoise the noise based on the noise distributed. And then using your model, you can slowly generate an image. And then pioneered by many groups, including An group and then Baker group, etc. We were also able to figure out how to do the same procedure on protein generations. Generally, in the Baker lab, the way looked like this. We started from the Rosetta fold, the structure prediction method and model, and then retrained the Rosetta fold to generate the Rosetta fold diffusion protein generation method. So what happened is like you start from Rosetta fold model, and then add the diffusion module to slowly add the noise towards the protein scaffold to learn the relationship between the coordinates distribution and the noise distribution. Of course, when you are doing the generation, you are reversing this procedure by slowly by starting from a group of noise, the distribution of noise, and then slowly denoise so that you can generate the protein with the distribution fitting your training data set, which is PDB. Working with Dr. David Jurgens, the method was continued to expand it to something what we refer to as C-alpha diffusion. This method incorporates the information of small molecule ligand and also change the frame to attention to C-alpha coordinate positions, and also added many other function modules. In the end, we were able to actually use this model to generate small molecule protein interaction using the diffusion method as well. Just give you some recent progress on the application of this method. One of the students I mentored, Long Chuan, he was able to directly take 3 ligands which share really high similarity to their structure, and then diffuse binders directly towards all 3 of them. With a success rate higher than 5%, he was able to identify nanomolar or even picomolar binders directly. Without any continual maturation of the binder, all the binders he identified were directly specific to their design target and do not have any cross-talking with the other target. This is just demonstrating how successful, robust, and validated our method is. As you may notice, the 3 structure he chose was structures pretty similar to fluorophores. So all these 3 ligands also have fluorophore signals. So we did something pretty interesting. So the student Long, he treated the 3 proteins as protein tags, and then tagged the proteins into different organelles to the cell, and then express them at the same time in the cell. And then he added all the 3 ligands. As you can see, in the cell, you can simultaneously light up all the 3 colors and then mapping all the different locations of the organelles. This is pretty remarkable because this is really helping and achieving the multiplex super resolution imaging, allowing in-depth like cell imaging studies for under more complicated scenario. Apart from the design, protein design and design method development, we do care about many real-world problems. So generally, as you may not realize at this point, my group and also the general protein design field works like this. So we have close collaborations and close communications with real-world application people, such as doctors, biologists, and industry folks. We focus on the phenotypes on what's a function to be useful to develop. And then protein designers work together, change and develop different models. And then these models will generate the proteins which we think would be achieving those functions. Because thanks to the nice expression and testing feature for the novel proteins, we try our best to develop automated and high-throughput assays that can allow fast verification of the design binders and also feedback the proteins generated data into the design model. And we push our molecules as fast as possible to our collaborators, allow imaging and allow real world problem solving in a relatively fast manner. So the aim of protein design is to just like create molecules in smarter ways, which is really helping for solving the difficult and challenging and valuable problems in the real world. So in my lab, I focus on some real world problem as well. First of all, the first direction, which I will also mention a little bit in my coming part of the talk, is we design binders and sensors to metabolites and ligands to sense the ligand landscape during disease progressing. And secondly, I also do many enzyme design type of project, such as expanding enzyme separate scope and also creating new enzymes which never existed in nature, so that we can allow new catalysis and reaction happening. And lastly, based on the functions we try to design, of course, there is a need and a function and also model improvement we will do in the group as well. Now just spend a little bit more time on the sensing. So for sensing problem, first of all, there is in vitro sensing. People probably have seen this glucose meter some diabetes patient wear on arm. This actually contains an enzyme that can oxidize glucose and then releasing hydrogen peroxide that can be measured to see the glucose level. First of all, many people still want the industry and folks still want better version of either sensor or enzyme to sense the glucose presence. Not only glucose, there are actually many drugs and metabolites, which is really also meaningful to detect in a real in-house manner. People don't really have to go to doctors or hospitals for this type of routine checks. These are referred to as in-house pollen care type of diagnosis. However, they are really limited by the fact that most of the ligands don't really have a well-behaving binder. Or if they have a native binder, those native binders probably need to be stored at 4 degrees C or minus 20 degrees C. They are just difficult to engineer. So de novo protein can really help to change this problem. On the other hand, not only in vitro sensing, we are looking for in vivo or tissue level of sensing as well. To just now understand more on how disease progress, for the past 10 years, there are lots of technologies invented. For example, Xiaowei Zhuang's lab, they figure out how do you mapping all the different cell types in tissue or even larger organelle level. And also we actually have pretty robust method to say, let's say if there's a target protein, we want to know how they are expressed in different time, in different type of cells, in different type of tissues. There are pretty good ways to say those as well. Here I'm going to show you a fake data. As you can see, if the question is, can we pinpoint where and what are the ligand presence on some different cells, for example, a cancer cell or early developed cancer tissue, etc. Can we just tell which are the ligands present there, and then what are the ligand correlation with the cancer with the drug with the disease development? There is fundamentally no such a technology right now that can solve this problem. By using de novo protein design, we can generate lots of different sensors and then actually really help to monitor the landscape of the ligand, and then really hopefully to solve this problem. Just to showcase and then say how we the preliminary data we have already achieved. So for de novo protein design, one thing we can really do is we design the protein into the shape and secondary structure and profile we want. We purposely, for example, design all our binder into these pseudocyclic structures so that we can simply by removing the loops, making them into ligand-induced oligomerization proteins. This is really good because they are so modular that you can put onto different modules of sensing proteins, such as nanopore sensors, or you can just like make the protein larger into bigger proteins so that you turn it into small molecule induced conformation changing proteins, or you slice it into halves to make it into small molecule induced dimerization proteins. And then once you have this small molecule induced conformation changing, oligomerization changing, you can connect the proteins with pretty modular split, let's say split fluorescence proteins. Then you just naturally have a sensing protein for this module, for this ligand of interest. Just give you an example on the sensing format. We have the split proteins there, and then when there's no analyte in the surrounding, you have a free pass of the electron signals, so they will have a stable readout as like the background curve. However, once you have a ligand binding to the protein, you will directly see a signal loss, which is suggesting presence of ligand. This is good because you can read out this electronic signal and couple this type of readout into wearable devices, etc. With that, I'd like to thank everyone, and then do let me know if you're interested in the lab or have any questions. I'm more than happy to like work with different people and then mentor different students, and then hopefully we can really collaborate, improve the method, and then generate more designs useful for the real world scenario. Thank you.